So continuing on from where we just left off, because I can't record more than 10 minutes at a time, let's go into the epigenetic implications of the DNA packaging around nucleosomes that we just went over. So, as well as a lack of extracellular signals, like in the case with the chicken erythroblasts, DNA may remain in its nucleosome-associated state as a result of epigenetic factors, which are factors that you can accumulate from your environment or you may be predisposed to from birth based on like nutrition that you received during pregnancy. So think stuff like the Dutch hunger if you want to look into that. But there are three main forms of epigenetic interference that will stop your free-floating DNA from being able to unwind itself from the nucleosome complexes. The first of these is methylation, where a methyl group essentially pins DNA to the nucleosome. So I think I have a picture of this from a later slide. These green things here would be the histones. Put them together and you call it a nucleosome. These yellow stringy things are the DNA, and these red methyl groups, you can see they look like pins. They go through the DNA and essentially kind of pin it to the histone so it can't unwind and the genes can't be transcribed. Then you have deacetylation, which is just removal of nuclear acetyl groups that prevents the DNA from unwinding from the histones due to differences in charge, which is a bit boring. And you have histone tail interference, which I believe is also shown on this diagram. See these little squiggly green things coming from the green balls? They are the histone tails. If they go through the DNA or they hold it in place, it essentially does the exact same thing as the methyl groups. It stops the DNA from unwinding from the histones from the nucleosome, and those genes can't be transcribed. So these factors prevent the unwinding of DNA into its free form, so it can't be transcribed or expressed. Epigenetics demonstrates how alterations in these expression of genes, because of these factors, can occur with no change to the genome, because the genome hasn't changed. You're just either adding methyl groups to it, you're deacetylating the histones, or the histones are poking through the DNA. There's been no change to the DNA, no change to the genome, but you're still seeing differences in the phenotype because certain genes can't be transcribed. And we'll get into a couple of examples of this in a minute, just to kind of better your understanding. So activators, like we mentioned in the last little lesson, they can increase the rate of transcription by activating an enzyme called histone acetyltransferase, shortly called HAT, which binds to upstream activating sequences of genes, catalyzing acetylation of histones. So the histones acetylation is increasing, and that causes DNA to unwind. And then on the other hand, you have repressors, which we mentioned before. They play a role in kind of inhibiting the effect of activators. And like we said before, these activator proteins are also responsible for creating a signal transduction pathway which leads to RNA polymerase, arriving at the promoter region of a coding gene and starting transcription. So the repressors can also activate an enzyme called histone deacetylase, which binds to upstream repressive sequences this time, causing deacetylation of histones. And when histones are deacetylated, it causes DNA to bind tighter around them, so the DNA remains wound around the histones in the nucleosome, so it can't be transcribed. So let's look at some examples of this in action. Quite a famous example, Morgan's white-eyed Drosophila mutation, which we can see at the bottom right here. See how one of them has a white eye and one of them has a red eye. The one with the white eye is not producing the red pigment, while the one with the healthy red eye is producing the red pigment. This mutation was not a result in changes to kind of the genome like you would normally think in mutation, where you have kind of insertions or deletions. It's the result of chromosomal translocation, which I believe happened as a result of crossing over during meiosis, where the gene that coded for the red eye pigment was translocated, so moved into a non-coding region of the genome that was not unwound from the nucleosome, so could not be transcribed. So we go back to that other image. You can imagine up here, you have the red eye pigment that sits here. So in a healthy one, it would be in between these kind of nucleosomes where it can still be transcribed, or in like the free floating DNA where it's been unwound, but it gets moved into a region of the genome that is not unwound when it needs to be. So the red pigment gene can't be transcribed by RNA polymerase, so none of the red eye pigment gets produced, which is why we see no red eye pigment in the white-eyed Drosophila. So this mutation is a result of transcriptional repression, like we've been talking about throughout this. Another very important example that I would recommend you learning quite well is the case of agouti mice, which are these fat yellow ones here. There's a healthy mice to the right, and the fat yellow one is the mouse with the mutated agouti gene. An experiment was done on them. So these fat mice, they usually suffer from having yellow fur, obesity, and increased tumor risk when the gene is transcriptionally active. Now, like we said before in the epigenetic causes for transcriptional repression, we can take that and we can forcefully transcriptionally repress this agouti gene by feeding these yellow mice a diet supplemented with bisphenol A, which is very rich in methyl groups. Mice on this diet display the healthy phenotype despite having the mutant agouti gene, so they should look like the yellow mouse on the left, but they look like the kind of blackish brown mouse on the right, because the agouti gene has been transcriptionally repressed as a result of these methyl groups pinning the gene to the histones and the nucleosome, preventing it from being transcribed. So despite having the mutant gene, it displays the healthy phenotype, but as a result of the diet that we give it, which is rich in the methyl groups, which methylate the gene, preventing it from being transcribed. In humans, pregnant women may take folic acid during pregnancy for kind of similar reasons, to prevent a birth defect called spina bifida in offspring. So that's all for here. Thank you.